بس أسأل طلابي IGCSE Biology 0610 شو هي أصعب سؤال بينسأل ببيبر 6، على طول بيكون الجواب Design an experiment أو Plan an investigation. هيدي البارت يلي بيجي عليها من 5 ل6 علامات ببيبر 6 كتير مهمة. بس أنا بهيدا الفيديو رح كون عم علمكم الخطوات والاستراتيجي لحتى تكونوا عم بتجاوبوا بشكل مفصل على هيدي الكويستشن، وتكونوا أكيد إنه تاخدوا الفول مارك فيها ببيبر 6. خليني انتقل أنا وياكم للشرح، وكمان رح نكون عم نعمل example عن هيدي البارت من الباست بيبر. خلينا نروح على الشرح.
So let's start. Design an experiment or plan an investigation. In order to design an experiment in paper 6, I want you to memorize IC Dr. S. Well, who is IC Dr. S? Actually, I don't know, but this will help you to remember the steps in order to explain how you can design an experiment and make sure that you are going to get the full mark. So I stands for the independent variable, and the independent variable means the variable that you are going to change in the experiment. And C stands for the controlled variables, or the variables that should be kept constant. Actually, all variables except the independent variables should be kept constant throughout the experiment. Don't worry, I'm going to show you an example in a few minutes. Well, D, it stands for the dependent variable, or the variable that you're going to measure. Okay? And keep in mind that you must always mention the method. So don't say only the dependent variable is blah blah blah, the independent. No, you have to say the method. Okay, so for example, if we're talking about investigating the effect of temperature, so you have to say, let's take for example 5 test tubes. Each tube is put at different temperature, so we're saying the method. And here also we don't say we are going to measure the breakdown of starch. You have to mention how you are going to measure the breakdown of starch. And now R in doctor is you have to say this sentence. Repeat the experiment many times, 2 or 3 times. Take an average and exclude anomalies. So this is 1 mark. You can always use it in your experiments. And finally, S stands for safety precautions, like wear a lab coat, tie your hair back, wear goggles. Okay? And then, if possible, you can mention the improvement. And one more time, the method of the experiment, the methodology is very important. Okay? So, IC Dr. S, independent variable, controlled variable, or what we are going to keep constant, dependent variable, what we are going to measure, repeat the experiment, take average and exclude anomalies, and the safety precautions. Now, let's make an example. So, plan an investigation to determine what the effect of different concentration of amylase. Okay, so this is the independent variable. This is what I'm going to change. Okay, on what? On the breakdown of starch. So the breakdown of starch is what I'm going to measure. It's the dependent variable. So let's start by IC Dr. S. So I is what you're going to change, or the independent variable, and the independent variable here is different concentrations of amylase. But please do not forget that you must mention the method, or what we also call here the replica. What do I mean by this? I mean that instead of saying the independent variable is different concentration of amylase, you have to say we are going to take 4 or 5 test tubes containing different concentration of amylase. And here we're done from the independent variable and the method of using independent variable. And then we're going to move to the controlled variable and make sure to mention at least 2 controlled variables. So everything, all the variables should be kept constant except for the independent or the different concentration of amylase. So what do you think we are going to keep constant here? Yes, exactly. So first of all, we have to keep constant the concentration of starch. We also have to keep constant the temperature because temperature affects the digestion. And see how we always mention the method. So when I say put that, instead of saying the tubes must be kept at a constant temperature, so say how you are going to keep the tubes at a constant temperature by using a thermostatically water bath. Okay, so this is a second controlled variable. Also, you have to put all test tube for a specific period of time. This is another controlled variable. And also, you can say use the same type of amylase. So this is another controlled variable. So make sure to mention at least 2 controlled variables, and we're done from the second step. Now, we are going to tell them how we are going to measure the dependent variable, how we are going to measure the amount of broken down starch. Okay, so think about it. If you have starch and the starch is broken down, so how do you know that the digestion of starch takes place? Yes, exactly. If this starch is converted to reducing sugar. And here we are going to use what? The Benedict solution in order to measure the amount of reducing sugar. So the dependent variable measure the concentration of produced reducing sugar. How? By using Benedict's solution. So every time you are going to take a sample from the 4 or 5 test tubes taken at the beginning, and let's say after 5 or 10 minutes, so after a very specific period of time, you take a sample and you add or you apply the Benedict solution test and you compare the colors. Okay, so here we're done from the dependent variable. Let's move to say the easiest part. Repeat the experiment to take an average and to exclude anomalies. And also another easy part is the safety precautions. So you have to wear goggles and tie your hair back. You can add also wear a lab coat. See, it's very easy. So I see Dr. S for independent variable and replica, the controlled variable or what we are going to keep constant, D for measuring the dependent variable and mention how you are going to measure it, repeat, and finally the safety precautions. Now I wanted to show you the mark scheme to see how the marks will be distributed on such type of questions. So on the method, you are going to, or they are going to give you 2 marks. So the method means how you are going to measure the dependent variable, how you are going to use the independent variable, so Benedict solution, water bath, thermostatically. So the method, 2 marks. The independent variable, 1 mark. Mention at least 2 controlled variable. Mention how you are going to measure the dependent variable. And finally, mention that safety precaution and say repeat to take an average and to exclude anomalies.
شو رأيكم؟ شفتوا إنه plan and investigation منا كتير صعبة قد ما أنتم فاكرين؟ هيدي وحدة من الاستراتيجيات يلي بعلمها لطلابي بال IGCSE Biology 0610 ب paper 6. كمان رح كون عم بختار بس 5 طلاب ينضموا لطلابي أنا بكوتشنج بروجرام اسمه Let's Crack Paper 6 لحتى أعلمهم كل شيء له علاقة ببيبر 6، كل الاستراتيجيز، ومش بس هيك نشتغل سوا بالباست بيبر لحتى أكون أنا أكيدة إنه رح يوصلوا على ال IGCSE exams مسكرين اخدين الفول مارك ببيبر 6. لا تقدروا تنضموا لنا لازم تحجزوا مكالمة استشارية مجانية لحتى أنا أشوف شو مشاكلكم وقد إيه في يساعدكم بهيدي البيبر. تحت هيدا الفيديو رح تلاقوا رابط حجز هيدي المكالمة الاستشارية المجانية. ما تنسوا، بس رح كون عم بختار 5 طلاب ينضموا لطلابي لحتى نشتغل سوا، سو ما تتأخروا بالحجز.